we are going to go to our main presentation, in which I'm sure that you're going to learn a lot of new things. We have Tim Weitzel, who is a natural resources biologist with the U.S. Fish and Wildlife Service and an expert on lamprey. And Tim was one of the experts who was consulted while drafting the wildlife impacts section of the Community Impacts Mitigation Plan. And it was really great to work with him again on this presentation, and we definitely learned even more about lamprey through this process. So Tim, at this time, I'll turn it over to you.
Thanks, Danny. Boy, it's going to be really hard to follow the quiz and questionnaire that you guys gave, so I'll do my best. And Danny, I am going to, just so I can try to drive from my end, I'm going to see if I can share my screen. Is that okay?
Yeah, sounds great.
Okay. Oh, this is, I can't share while you're sharing. Can you disable and then I'll try?
Yes. There you go. You should be good to go.
Okay. Okay. Oops. Okay, so all words are seeing a full-on slide and presentation mode, correct?
Right now we're seeing the presenter mode. If you can go up to display settings at the top, it should switch. There we go. Yes.
Got it? Okay, great. So again, thanks for the introduction and thanks for the opportunity. I appreciate everybody's time. As Danny said, my name is Tim Weitzel. I work for the U.S. Fish and Wildlife Service. And we're going to go through a little bit of information, hopefully about lamprey. I can't see folks the way my screens are set up. I apologize for that. So if there are any questions, just jump right in. All right. So I wanted to start just a little bit with some background and some information on evolution relative to lamprey. And some folks may have seen many of these slides before, but in this case, we think of lamprey as living fossils. And so basically what you see in this slide is a timeline with lamprey on the far left. And so they show up in the fossil records about 450 million years ago. So they've been around for a long time. And for context, if you sort of go to the right on that red timeline, you see that dinosaurs first appeared in the fossil record about 250 million years ago. And then dinosaurs disappeared about 50 million years ago. So lamprey were around a long time before dinosaurs to watch dinosaurs come, watch the dinosaurs leave, and lamprey are still here. And then on the far right of that timeline, you can see that about 6 million years ago is when we first started seeing salmon in the fossil record. And then not very long ago, in a relative sense, humans showed up. So they basically just get a little line on that time scale. So it gives you some sense of how long lamprey have been around. Partly because of how long they've been around, they've diversified into a number of different species. And this is a very active field in the lamprey world. Species name, how things are related, is studied pretty intensively and can change fairly frequently. But where we are right now, there are about 41 species worldwide that have been classified or designated. This is a list of most of those. It's not all 41, but it's most of those. It's just a simple dendrogram showing their genetic relationship. And the reason I put it up is because I thought it might be useful for people to know. So in the case of Portland and the Portland Harbor specifically, there are 4 on this list that you can find. The first little lightning bolt that you see in the middle of the screen is Entasphenus tridentatus, otherwise known as Pacific lamprey. And please note that their genus is Entasphenus. And then the 3 at the bottom are Lampetra reesii, Lampetra richardsoni, and Lampetra pacifica. And we'll talk some more about those, but those are the other 3 species that you could find in Portland Harbor And again, note those are all in the genus Lampetra. Let's see, here we go. All right, so thinking about those 4 species that you might find if you were out playing around in Portland Harbor. This is just a very simple diagram. Thought I'd walk us through the Pacific lamprey life cycle that most folks probably know, but just wanted to make sure we were all sort of at the same level. And so I'm going to start kind of at 2 o'clock in that square that's outlined in pink. So adult Pacific lamprey will spawn in freshwater, in gravel. They nest in stream riffles, and they die after they spawn. So we call that semelparous. They spawn once and then they die. So they're in freshwater, up in tributaries, spawn, then die. So if you go, we're going clockwise now. If you move down, you'll see the embryo developing. And so the embryo hatches into larvae. And then the larvae, which are also called amicetes, drift downstream, sometimes a little ways, sometimes not very far. Basically, once they hatch, they emerge from the nest. They're trying to find areas to settle out, and they burrow into the substrate. So they live, continuing clockwise, they live in the sand and silt. Some things to note about the larvae: they're blind. Again, they're burrowed. They filter feed. So unlike what a lot of people think of when they think of lamprey and being parasitic, sucking blood and all of that kind of business, these guys are literally sitting in the sediment. They are spitting some water and they're sucking it down into their mouth, and they're filter feeding on plankton and various things. So they will be in freshwater for, I mean, what we've learned now is probably it's more like 2 to 12 years. Can really vary. And then at some point during that time period, they go through a larval transformation or metamorphosis where they transform into juveniles, which are also called macrothelmia. Those juveniles, so it's a true metamorphosis. So now they develop eyes, which you can see in the photo as we're moving clockwise. They develop teeth, so they're no longer filter feeders. They have organ rearrangement, so it's a true metamorphosis. If you know, the most common way to think of it is like butterflies, right? I mean, it's a true rearrangement of tissues. The macrothelmia or the juveniles are in freshwater, likely for anywhere from a couple of months to a little over a year. But eventually they make their way and migrate out to the ocean. They're in the ocean for, this slide's just 2 to 3 years. Again, as we're learning more, it's probably more like 2 to 5, maybe even 2 to 6, that they're out there. They are parasitic. They're feeding on host fish. They attach and they're feeding. In the image that I skipped over, typically it's the tooth pattern on the sucking disc that's used for identification once they transform. So in this case, Pacific lamprey are called Entasphenus tridentatus. And so if we had a big blow up and we were doing some species identification, you would see 3 primary teeth, which is why they're called tridentatus. So after they're in the ocean for that variable period of time, something triggers them. They become reproductive. They enter freshwater streams again. They come back in and they go to spawn in tributaries to start the cycle over. One thing to note about that part of their life cycle, yes, they're anadromous, right? So they go freshwater to saltwater, back to freshwater. But unlike salmon, which is what most people are familiar with, they do not home to natal streams. And that has some impact on their population structuring. All right, so quick and dirty on Pacific lamprey, their life history cycle. Again, summarizing, they're anadromous, freshwater, saltwater, back to freshwater. They're parasitic, so once they transform, they attach and feed off bodily fluids of their hosts. They have low population structure because they don't home. The adults end up, and this is a rough range, but 70 to 90 centimeters. And again, they're semelparous. They spawn once and then they die. All right, so now we're thinking about those other species that were on that list. So western brook lamprey, remember now we're now in the genus Lampetra. They are resident, so they are not anadromous, never leave freshwater. They're non-parasitic, so they don't attach to host organisms and feed on bodily fluids. They filter feed as larvae, and then when they go through the transformation, they become adults ready to spawn. They have relatively high population structure. So a way to think about that is, again, I'm just going to use salmon as a reference because lots of folks have heard about salmon. Various different populations of salmon across the Pacific Northwest. A lot of that has been defined based on their genetic relationships. Western brook lamprey, and a lot of these are average figures, but on average are about 10 times more structured or intermingle 10 times less than Pacific salmon do. So the brook lamprey have high population structure. Maximum size of adults is roughly 12 to 18 centimeters. And again, they're semiparous, spawn once and die. Western river lamprey. So this is the Lampetra oryzii, third species on that list. They're anadromous, so freshwater, saltwater back to freshwater. However, their time in saltwater is weeks, maybe 10 weeks. They don't go very far. They are parasitic when they're there, again, but not for very long. They have relatively high population structure because they don't go very far. They end up tending to come back to where they were from. Maximum size is roughly 15 to 25 centimeters. And again, semiparous, spawn once and die. Let's see. So quick note, you saw on that dendrogram that there are many, many species of lamprey. So Western river lamprey, or Lampetra oryzii, has been designated as a species, and it's thought of as a paired species with Western brook lamprey. So one's resident, one is anadromous. However, recent information suggests that we really can't tell them apart genetically. And so now the thinking is becoming more that they're an ecotype as opposed to two distinct species. And the simplest way for me to think about that is, again, using the salmon steelhead world. If I think of a steelhead trout and a rainbow trout, genetically they're very similar. One expresses anadromy, one stays as a resident. Similar concept. And then the final of the four species on that list is Pacific brook lamprey. Again, some of the summary characteristics, it's also a resident, so freshwater only, non-parasitic, filter feeds, and then transforms into an adult. We assume that it has high population structure, although there's not a lot of information on that. Maximum size is roughly 20 to 25 centimeters as an adult. Semiparous, spawn once and die. So again, thinking of species, you saw it, it's got its own name on the list, Lampetra pacifica. However, recent discussions in the literature talk about whether these are truly a unique species or a synonym, which is really what most people are thinking at this time. And by synonym, what I mean is it's essentially the same species as Western brook lamprey, but for whatever reason, there's this group of lamprey, these Pacific brook lamprey, that tend to have fewer trunk myomeres. And in that image, you can see the pink lines. So myomeres are basically just those muscle fibers. So they have fewer trunk myomeres than a typical Western brook lamprey. And these Pacific brook lamprey also tend to be found in coastal areas. All right, so a little bit of quick and dirty information on lamprey as a whole. Now, I was going to spend some time just focusing on the harbor-wide monitoring that we've been involved in relative to Superfund activities. A couple of quick things before we get into some methods and results stuff. We've done a bunch of monitoring over the years. There are lots of people that have contributed. Putting them all on a list doesn't do them service, but here is a list of some of them. You might know some of them. Some of them I think are actually on this call. And we are eternally grateful to everybody for their help in all of the work that you're about to hear about. Equally, there's funding been provided by a number of different entities, and we wanted to acknowledge folks who have contributed financially to try and help get this work done. All right, so we are doing some monitoring relative to lamprey in the harbor, Portland Harbor. So specific questions that we have been trying to answer, and they're relatively straightforward, I think. There are four of them. Do larval lamprey occupy the lower Willamette River, LDR, LWR? Are they there? What is the probability of detecting larval lamprey in the lower Willamette River? So once we found them, and we wanted to know how well we could go collect them or detect them, how easily we could detect them. Are they distributed throughout the lower Willamette River, or are they clustered in certain areas that we can sort of differentiate? And do multiple size, and we sort of use size to infer age, although that's not a perfect relationship. But do multiple size classes of larval lamprey occupy the lower Willamette River? Or is it just one group that's going through one age class? So those are the four questions that we've tried to focus on. A little bit of information about our approach. So the sample framework that we've used. So everybody probably on this call is familiar with the Portland Harbor area. So we have worked basically from Willamette Falls down to the Columbia River. So that includes Multnomah Channel. We took that entire area and we divided it up into 30 by 30 meter quadrats. So you see these little boxes. And this is just an example of a section of the river that got divided up into these quadrats. Each of those got assigned a geographically referenced center point, and this is all to set up our sample design. And then we use this random spatially balanced ordered approach, what's called a GRITS approach, to select sample sites. And basically what we were trying to do there is make sure that when we went out and looked for them, we weren't biasing for better or worse where we were sampling when we were trying to look for these fish. In the end, we ended up with 84 quadrats that we targeted. I didn't go into why we chose 84, but based on things like detection probability, we were able to sort of think about the effort we needed to put in to try and answer some of the questions that we had. And it ended up being 84. And again, they're in priority order. A quick note. So we are doing what we would refer to as a BDOCI. And that official acronym stands for before, during, and after control and impact assessment. In this case, it really translates to pre-restoration, and during restoration, and post-restoration assessment. And that's the site-specific stuff that's being restored, not in the harbor specifically. All right, so a little bit about the method. That was the framework, a little bit about the method. So we use, because we're out in this big river, we use a deep water larval, a lamprey electrofisher that we got from some colleagues and did some modifications from some colleagues in the Great Lakes area. A quick note, the sample area, every time we drop this down and sample with it, we're sampling less than 1 meter squared or less than 1 yard squared, depending on what you're most comfortable with. And you'll see some images here in a minute. So this is the apparatus that we use to generate some electricity for this electrofisher. That attached by some wiring, you can see an orange wire there that runs along this boom. So it runs from the generating unit, runs along this boom to what we call a bell. So you can see that this biologist is grabbing onto the bell, so that area at the end. So those wires that run to the bell, so we can send an electric current down to the bell. It's that electric current that it's kind of like static electricity, if you've ever sort of rubbed your feet on a carpet and then shocked somebody. And it irritates the larvae who are burrowed in the sediment to come out. They come out and they're underneath this bell, and then we apply some suction that goes through that bigger tube that you can see. And that suction, anything that emerges that's under the bell, we're sucking everything, that's there up through this tube, and it gets filtered through this basket. And so that's how we capture larvae if they're there. And so then a quick note too, because I've already pointed out there are 4 species that represent 2 genera that potentially are there. And so we typically use tail morphology or caudal morphology to help us identify the larvae. The specific note I wanted to make there is really if they are shorter than 60 to 70 millimeters long, you can't identify them reliably. Morphologically, you can only do it through genetics. So sometimes we get very small ones that we just know are larval lamprey, but we don't know what species or genus they came from. All right, so some results. So this is the before. all the site-specific restoration started. So this is some stuff we did in 2007 to 2010. Again, a picture of the lower Willamette River. In this case, what you can see is we've divided it up for sample and question purposes. We've divided it up into 4 subreaches. We've got the upstream subreach, Willamette Falls down, and then we've got the Superfund proper area. We've got the lower Willamette, and then we've got Multnomah Channel. So 4 subreaches. So we did a bunch of sampling. And this is just a quick summary of that sampling. But really what you can see from this table is that we found lamprey, larval lamprey, in a variety of seasons, spring, summer, fall. We didn't happen to sample in the winter. We found both genuses were represented. So you can see, I'm sorry I don't have an arrow, but you can see that second and third to the last column, you see PCL, so those are Pacific lamprey, and Lampetra species, so those 3 species that you can't really tell apart. You can see that we found both, and we also found some unidentified ones, so those are ones that were too small. We found them everywhere we looked except Multnomah Channel, did not find any in Multnomah Channel, and our overall detection probability was 0.07. Which basically means if we drop that bell 100 times, we would detect lamprey 7 times. Some other results that were meaningful to us, we stratified the area that we sampled by depth, and there was no significant difference. We looked at less than 5 meters, 5 to 10 meters, and then greater than 10 meters. I'm sorry about that. And basically, our detection probability was the same in all. So there didn't seem to be any sort of stratification by depth. We also saw multiple length, and maybe, again, you can hopefully infer a little bit age classes. So we saw very little ones, 30 to 45 millimeters, up to, relatively large, 110 to 125. So lots of size, lots of age, potentially classes are represented in this area. If anybody's interested in more detail, that was super quick and dirty, but we published the work. This is just a reference to the publication. I'm also happy to share that. So a quick summary. I think this is kind of interesting in some ways for context. So that whole lower Willamette River that you've seen now, when we divided it up into those 30 by 30 meter quadrats, there were over 31,000 of them. It represented over 28 million meters squared. We sampled, remember we only went to 84 quadrats and we only dropped the bell once. We sampled just under 35 meters squared, and we found larval lamprey. We found them in 3 of the 4 subreaches, all depths, and basically all size classes. And our site-specific probability of detection was 0.07. You see some confidence around that. And I just like to highlight that, and this is the way I typically put it. Lots of people are like, well, you didn't find many lampreys, so what's the big deal? I like to sort of flip that on its head. So we only sampled 0.000123%, and that's correct. It's 0.000123% of the entire area of the lower Willamette, and we found lamprey. So in some ways, I think it's actually amazing that we found lamprey, as opposed to no big deal. So next steps, again, still talking about harbor-wide monitoring. So this is what's going on right now. So the design we're using, we're currently involved in it. The design we're using generally follows the Jolly et al. Or the pre-work. There are those 4 subreaches from the Columbia River to Willamette Falls. And we formally added in seasonality. We're doing 2 seasons, early fall and early spring. Let's see. So we're scheduled to implement this harbor-wide monitoring between 2023 and 2025. So obviously fall of 2024 and spring of 2025 have come and gone, and we were able to sample during those periods. In terms of results, so for fall of 2024, we went out in October, went to those 4 subreaches, ended up sampling 86 quadrats, so at least 21 in each of the subreaches. And for larval detection, downstream, we found lamprey in 3 of the 21, or 14.3%. And for reference, the 3 of 42 is what we found in that pre-work. So you get an idea, and it's just one season, 1 year. So I wouldn't put too much stock in any of this, but I wanted to give you some context. So in the Superfund site specifically, in the pre-work during the fall, we did not sample that, so I have no comparison. But last year we sampled and we found lamprey in 0 of the 21 quadrats that we went to. Upstream 0 of 23, and again, we did not sample upstream in the fall during our pre-work. And then in Multnomah Channel this past fall, we found lamprey or detected lamprey in 1 of the 21 quadrats, 4.8%. And again, previously it was 0. So if you sum it all up, we detected lamprey in the fall of 2024 in 4.6 quadrats though, for 4.7%. Previously, it was 3 of 63 for 4.8. So really pretty similar numbers. And again, one sample period in this during phase, not putting a whole lot of stock, but just for some context. All right. So spring of 2025 just happened. We went out in April, 4 subreaches, 86 quadrats. Larval detection, yes. It's really hot off the press, and I haven't had a chance to summarize that data. But we did go out this past spring. We did find them. We found them in more quadrats than we did in the fall. And that's really about all I have summarized at this point for the spring work. So again, one last thing about context that I thought was relevant. Some folks that we work with have published some stuff, and a lot of what we think about is there's a ton of seasonal, annual, and density-related variance. So it's really important to keep that in mind as folks, think about how to interpret all of this. All right, remediation. We're getting to the end, folks. Stick with me. Harbor-wide remediation. So the question we get a lot is, what's good for lamprey? Well, there's limited scientifically controlled, what we would call preference experiments. So I can't tell you with great certainty that, hey, larval lamprey prefer X. Go do this. So there's really no silver bullet that I can bring to the table. I know that's maybe disappointing, but it's the most honest me that you can get. But what we do instead, because it's not been well studied, is we try to consider this kind of question in the context of the life history of these critters that we sort of just went through. And so really briefly, starting with the embryo life history stage, we know that there are some examples of lamprey spawning and embryos developing in 5th order streams, a little bit smaller than the Portland Harbor. It's conceivable that they could spawn somewhere around the Portland Harbor, but it's really not something that we know much about. We don't really think it happens very often, if at all. And so I don't think there's much that can be done relative to embryos, at least not at the moment, given what we know. Larvae, thinking of their life history. Well, they're blind, they filter feed, and they burrow. They like to burrow so they can hide. They like to burrow because they're not very good swimmers and they need to get out of the current. So providing them, or ensuring, not necessarily providing, but ensuring that there's some substrate they can burrow in probably would be useful. Juveniles. Juveniles are migratory. So we know that they can become impeded by various things in migratory corridor. So making sure the migratory corridor is available seems like a good idea. Adults, also migratory. Providing them a migratory corridor also seems reasonable and appropriate. Hopefully this video will play for you guys. Danny, is that playing?
Yes, that is playing.
Okay. So these are adult lamprey. Envision them climbing up Willamette Falls. They can suck and climb. The problem is right here. When they hit a right angle, they can't get over that right angle and they fall. So they can climb very great distances if there's a wetted surface. But if there's a right angle at the top, they can't get over that because of the mechanism they're using to climb. So when you think of dams, when you think of fish passage structures, making sure that they don't have those right angles to deal with is probably a useful consideration. Additional things that might be important, and again, we don't know a ton. Contaminants, there's some examples of contaminants inhibiting or impeding larval burrowing behavior. We're not quite sure how it translates to the population level. Temperature, there's not a whole lot of evidence that warm temperatures is bad for larvae. There's not a lot known about juveniles. There's some indication that really extremely warm temperatures might be bad for adults. I mean, so there may be other considerations, but again, just trying to think of it relative to their life history. And then the final sort of thing to leave you guys with are in terms of possible actions, we, and I guess I should say but I'm going to say we have started to really think about these in terms of, we break them down into kind of three categories. One is to do, one is to undo, and one is to not do. And by to do, I mean things like, go dump a bunch of sand in Portland Harbor. That would be doing. Undoing would be, hey, there was a culvert. I'm taking the culvert out. I'm not going to do anything else. I'm just going to remove something that we did previously. And then obviously not doing is just to leave things as they are. And the reason that we sort of have started to view them this way is, we throw out this cautionary tale. On the one hand, you provide a bunch of sediment. In theory, it would be really good for larvae, right? There's a place to burrow, hide, filter feed, and so on and so forth. On the other hand, it can be such a good place that it ends up attracting a lot of predators. This is a picture of the stomach of a gull that's full of lamprey, larval and juvenile lamprey. So sometimes we know, sometimes we don't know. And if we don't know, maybe the best thing to do is just leave things as they are. And finally, it's when in doubt. Again, they've been here for 450 million years. So if we stay out of the way, there is some evidence anyway that they can figure things out. All right, and that's what I have. Right about a half an hour, I think. And I'm very happy to answer questions or take any feedback y'all have.
Great. Thank you so much, Tim, for that awesome presentation. And we do have a lot of questions in the chat. So we will start with the question from Cassie that was the first in the chat. And it looks like that's a question that should go over to Hunter with EPA. Hunter,
Y-
you're
Yeah,
on.
Danny, do you mind reading the question for me?
Yes. So the question is if lamprey monocetes can be found, it can be in Portland Harbor sediment for 12 years, does the rod and designs take into account that length of time?
Well, so the Portland Harbor cleanup is expected to take 13 years. So, that's a very, very interesting how those kind of line up. But it's not specifically designed around the life cycle of lamprey. And, I don't think that I would say I know that the remedy did not really use lamprey life cycles as a decision point in any of the deciding of the kind of how the cleanup plan was designed. That being said, we've considered lamprey in our biological assessment and are thinking about how we can best implement measures to reduce impacts on lamprey. But sort of as Tim's kind of touched on there, we are finding ourselves rather limited in what we can do to really minimize the disturbance of lamprey, amicetes in particular, from the remedy. Seems to be sort of a lesser of evils issue. And I think to me, what I take away from a lot of this is the best thing for the lamprey is to get the cleanup done as soon as possible, and to remove the contaminants from the environment.
Great. Thank you, Hunter. Next question we have is from Emil for Tim, which is, what are the chances of larval lamprey getting out of the way of a cleanup effort and dredging? And what about the process of cementifying the sediment in quadrants? How would that affect them?
I wish I could say with certainty. I think based on everything that we've seen, and this is pure speculation, we haven't really studied that and sort of quantified the chances. That's the question asked. But, I think there is a chance that, and it seems likely that some would get up out of the sediment, some wouldn't. whether it's during dredging or whether it's during cementing. And obviously, cementing is different in some ways than dredging, but it's really hard to understand the population impacts because, for example, in dredging, if we were able to stimulate them to come out of the sediment, it might be good that they don't get dredged, but then they might become food for bass that are swimming around. So um, I don't really know how else to answer that question. There's a chance that some will get out. I don't know if it's a good thing or a bad thing.
Great. Thank you, Tim. And then two related questions um, about sampling in the Portland Harbor Superfund site area. Is that something that has been done or is planning on being done in the future?
Yeah. So we sampled. So if you are you talking specifically about the Superfund, the formal Superfund area?
Yeah, the formal Superfund area.
Yeah. So we, in the pre-work, we sampled in the formal Superfund area. We just didn't happen to sample there in the fall. It wasn't associated with this particular project. We're actually taking advantage of the fact that we had done some work before this all started to happen. So we didn't know we should have been sampling in the fall in that area. In the Since the cleanup and the remediation has started, um, we started in the harbor wide again this past fall. We will do fall, spring, fall, spring, fall, spring for 3 years. And then at the end of this sort of 20, 25 yearish cycle for the remediation activities, the site specific activities, we're scheduled to do fall, spring, fall, spring, fall, spring again. So we should be in the formal Superfund site 6 times. Well, yeah, I guess 12 times, right? If you count both seasons um, between now and then 10 years from now.
Great. Thank you. And then we have a question from Michael Pouncil. Has there ever been tracking placed on lamprey? And if so, where have they been tracked to geographically?
Um, is there any way you can tell me if there's a specific life stage that you're referring to?
Yeah, Michael, do you want to pop in the chat or come off mute uh, if you have a specific life stage in mind? We'll see.
Yeah, and I
Uh,
mean I can answer it broadly. But there are so many life stages. So um, adults have definitely been tracked. Typically, what's happened is they've been given pit tags, a certain type of internal tag, or radio tags, and they've been tracked once they return to spawn. So it's really just tracking their movement once they're back in a stream. Not so much out in the very difficult. Um, Larvae and juveniles have been given those internal tags while they're in freshwater, typically tracking their movements out of freshwater or while they're in freshwater. Once they're in the ocean, they become pretty difficult. The problem becomes you can't put enough tags in to get a large enough return. It's a sample size issue. So once they're in the ocean, we have sort of less information, but we do know from genetics and other things that they distribute I mean, you find Pacific lamprey from Alaska to northern Mexico. Um, so I don't know if that's a short answer, I guess, to a potentially long question.
yeah, thank you. We have a question from Jan. Can tribal knowledge help us understand or teach us what lamprey need to inform remediation?
I sure would think so. I mean, and the more we can learn and understand, then the better. It's just on how you take that knowledge and think about it.
Great, thanks. And this is a question directed to EPA. So can EPA commit to ensuring research on lamprey post Superfund cleanup?
Yeah, and it's not a very satisfying answer, but I think the answer really is no. But that's just because we don't have pre sampling. We haven't been sampling lamprey. EPA It's just really not part of the cleanup, and that's not because we don't want it to be. It's just we don't really have teeth to require lamprey. They're not an endangered species or a threatened species, nor are they a species that we use to monitor for contamination for fish advisories or anything at this time. That being said, the reason is the mechanisms being monitored for now is just through other means. Tim and his folks are monitoring it, not because EPA is saying to do it. They have their own reasons for monitoring it. And so it's not to say that lamprey won't be monitored forever and into the future. It's just that EPA isn't doing as part of our Superfund cleanup. But I would assume that lamprey would be something studied, um, you know, forever in the Lower Willamette River, is just as a culturally significant resource.
Great. Thank you, Hunter. And a question going back to Tim from Laura Feldman. I found larval lamprey in the Columbia Slough at Smith Lake. How does the slough figure into successful migration?
So you said larval, is that correct, Annie?
Yeah, larval.
Yeah. Yeah, I mean, again, lots of unsatisfying things because they're so, we don't know a lot. I'm not exactly sure how it figures into it, but, the story that we've started to tell ourselves, and we try to always pinch ourselves to make sure we don't fall in love with our stories, but, these fish are spawning, these adults are spawning in these tributaries, and then they're there for, let's say, 2 to 12 years. They don't swim very well, but they do get up out of their burrows. And so just envision them sort of leapfrogging over time, washing downstream or floating downstream or whatever until they find the next place to burrow. So you find them, a lot of them potentially in Portland Harbor, including Columbia Slough. So, how significant is that as they're sort of working their way down and around and through Portland Harbor? I don't know. But it's habitat that they use. So that makes it significant at some level.
Great. Thank you. And the last question we have in the chat, your results slide showed some class sizes were not represented. Is this evidence that those size classes are absent from the population, specifically the 94 to 109 size class, or is this possibly related to sampling methods, design, or sample size?
Yeah, I think it's related to sample size, and so that information again was sort of the pre-work that we did. Since then, we've done a lot of other work, and we found since then we're able to find larvae that are as small as I think 11 or 14 millimeters, all the way up to 120 something 130 maybe. So basically, all size classes from what we would think they would be when they hatch to what we would typically think of as the largest larvae before they're going to transform. All the size classes seem to be there.
Great, thank you. And we actually have um, two more questions. One is a follow-up from Jan, which is regarding learning from tribes about remediation. Um, are the tribes involved or have they been consulted regarding lamprey and remediation? And this might be a question for Tim and Hunter.
Yeah, Hunter, I'm going to let you take that or somebody else because I don't know who's been consulted specifically from a remediation perspective. They definitely are consulted from a monitoring perspective.
Yeah, and as well with remediation, we have a technical coordinating team with 6 tribes involved, and we run all of our technical decisions by them. And many of them are present here today, or some of the representatives and consultants are present here on the call today.
Great. And a follow-up from Dwight. Was the survey that you were and are involved in, Tim, funded at all by the Portland Harbor Natural Resource Trustees? I know you had a list of funders on one of your slides.
Yeah, so my understanding of the mechanism of how it works is the trustees aren't necessarily the funding agents, but they are the ones sort of the liaisons between us and the funding agents. So the responsible parties are the ones that are actually contributing the money plus the Fish and Wildlife Service.
Great. And then a point of clarification, um, were larvae encountered during any surveys of the Superfund area?
Yes,
Yes. Okay, great.
100%.
Great. We'll give a minute for any last questions in the chat. Is there anybody who wants to come off mute to ask a question verbally?
And I apologize. I know this is going long.
No worries. A lot of great content here. Yeah, I see Doug's hand.
Okay, my understanding from what you're explaining is the lamprey only burrow in soft bottom. Would you expect them to find in river rock or cobble beds?
Yes, but it's sort of a nuanced answer. So mostly coming from our friends in the Great Lakes, there has for larval habitat, it has been classified into what's called type 1, type 2, and type 3. Type 1 habitat is basically sand, thick sand. Type 2 is the cobble, gravel cobble, and type 3 is more like bedrock. You can find them in all three. And the nuance is basically, let's take the extreme. If you have bedrock that has crevices and sand builds up in those crevices, you can find larvae burrowed in that sand. So even though overall it would be classified as type 3 habitat, you can find larvae there. Now, do you find them as abundantly? Typically not. But point being, if there is a place for them to burrow and they're there around, you can find them in all of those habitats.
One of the PRPs in their remedial design was proposing at the end of their cleanup to put river rock along the shoreline. So that would be counterproductive for larvae for the lamprey. It sounds.
It would likely be less optimal for them to burrow. Yeah, correct.
Thank you.
Yep.
Great. And we'll take one last question. I see Cassie's hand.
Thanks for this, Tim. Two to your question. One is sounds like EPA cannot fund sampling post active cleanup of the Superfund site. Is Fish and Wildlife, are you interested after that 13 years of cleanup to see like if there's a increase of populations in the Superfund area specifically, and would Fish and Wildlife commit to that, like documenting that, researching that?
Well, the simple answer is we're definitely interested, right? So we started doing this work in 2007 because we were being told that main stem areas were just pass through migratory corridors for juveniles and adults. We weren't sure that was correct, so we started doing the work in 2007 and found larvae. And since then, we've been interested for sure. For the Superfund stuff that's going on, these site-specific restorations, we have been involved since it started. Obviously, are currently involved, and roughly speaking, are classifying us as being sort of halfway through the process. And then technically, we're supposed to continue sampling. So we're supposed to sample 1 year pre. So pick a restoration site. Sorry, maybe this is a more direct answer. Let's think Alder Point. So we were supposed to, what we're committed to is sampling 1 year pre, and then years 1, 2, 3, 4, and 5, 10, 15, and 20 post. For all of the site-specific restorations, and they all came online at slightly different times. So all in all, we sort of went out before this all started, and then we're committed. We have committed to be involved for about 20 years, 25 years. All of that being said, I'm just going to be frank about it. I work for the federal government, and I can't really commit to anything at this point.
Mm-hmm. Okay.
But we're interested.
Thank you. And if we have And one more quick question about, you know, if parties happen to be proposing putting some mid-level Portland Harbor sediment at Ross Island, around Ross Island, filling in around it, how would that affect amicetes?
Well, again, I mean, unsatisfying. No, I can't perfectly know that, but my guess would be, if there is good sediment, and by good, I mean, uncontaminated, anyway, I mean, good, sediment that the larvae can burrow in, they likely will. I mean, there clearly are fish around that area. Ross Island is one of our reference sites, and we know that there are lamprey there. So I suspect that if there were more places for them to burrow, they would burrow because they're there. What that means, again, I don't know. Is that a good thing or is that a bad thing? Does that keep them from leaving when they should leave? I don't know all of those answers, but I am suspecting they would burrow. And a good, an example, I don't know if it's a good example, but for any folks who are familiar with Leeburg Dam, it hadn't been sort of maintained in quite a while. There was a large buildup of sediment behind the dam. And there were tons of, I mean, it's not a technical term, but there were tons of larval lamprey in that sediment. So they ended up there. I would suspect the same thing would happen.
Great. Well, that's all the questions that we have time for. So
Danny, do you mind if I just jump
yeah.
in for a sec? I feel like I want Tim to clarify something because I've seen some kind of things pop up in the chat. So I'd really like Tim to really clarify because I'm seeing this in the chat. Did you find larval lamprey in the Portland Harbor Superfund site area?
Yeah, we have found larval lamprey in the formal Portland Harbor Superfund site area. Yes.
Yeah. Thank you, Tim.
Mm-hmm.
Yeah, thanks for that clarification. And thank you so much, Tim, for your amazing presentation and thoughtful answers to all the questions. We really appreciate you taking the time and putting together this great information for us.
Yeah, thank you guys. Keep doing great work.
Thanks.